I have already introduced you to the basic structure of the ear and eyes. So, in this lecture what I will try to do is that I will just go do a recap in the form, like initially when I showed you, I just drew all the figures of the hair cells or the rods and the cones. So, in this lecture I will give you the actual picture, like I have collected all these from different journals, and I will give you all the references. Please go through them, and eventually we will be talking about how this hearing prosthesis or the cochlear implants or the camera in front of the eyes are being fit. Okay. So, to start off with, so there will be two classes which I will be taking consecutively. This will be the section of the implants, man machine interface and the implants and all those things. Okay. So, the title of this class is Biology of Hearing and the Cochlear Implant. This is part 1. There will be then eventually we will move on to the vision, and then we will move on to the brain. Okay. So, this is the reference I wish all of you to kind of consult, the Cell Biology of Hearing by Martin Schwendler and Kasher and Muller. This was published in Journal of Cell Biology in the volume 190, part 1. Rather issue 1 from page 9 to 20. Kindly go through this. This is available online. So, this will be a big help to understand. So, this following paper, the Cell Biology of Hearing by Martin Schwendler and Kasher and Muller, which is published in Journal of Cell Biology in the volume 190, issue number 1 in the year 2010, will be a big aid to understand some of the structures which I have already discussed with you in one of the classes. So, here I want you to go through it. Okay. So, coming back to the structure of the auditory sense organ. Okay. So, if you look at this structure, you will find that this is where what I have already showed you, that the outer ear. Okay. You could see the outer ear out here, and then you see the inner ear. So, the middle ear and all the 3 bones, and here is the inner ear, and I told you that there are hair cells which are present. So, these two micrographs what you see out here underneath, there are 4 pictures out in this picture. These 2 pictures, this one and this one, you could see these are the arrangement of the hair cells, which is shown like, one second, let me, okay. So, they are in array and they are actually sitting out here inside the cochlea, what I have drawn. Okay. Now, if you look at this picture at a further resolution, they are if you kind of see the dimensions of the individual cells, that will give you an idea how small these structures are which are sensing your different frequencies. And this reference, again, this is another reference which I want you people to go through, is by Furness, D. and Furness in Journal of Physiology in the year 2010, which was in the issue 588. Okay, please go through these things where you have a scanning electron microscopic image of hair cell bundles with the inner hair IHC inner hair cells here from the part of the rat postnatal day 10. Okay, please go through this, and that will kind of give you an idea at these are the nano machines of the body which helps us to decipher wide range of signal from sound, light, smell, taste, so on and so forth, touch. So, likewise, so these are those specialized structures. That is the reason why I am taking this special lecture, where I wanted to you guys to expose to those specialized structure which are translating the physical signal of nature into electrical signals. These are really some of the profound machines which has evolved over the course of evolution of thousands and thousands of years. Okay. So, now look at this one. If it moves in one direction, then it will pull all of them in one direction, and I will show you that. So, if you look at it, they are arranged in a height as if like in a staircase, they are arranged in a staircase. If you look at the slide now with this, so look at this, they are almost arranged like a staircase model out here. Okay. So, they are all like, so now if you look at this cartoon what has been drawn out of those pictures, So, it is as if you are sitting in a photo session. Look at this picture, as if these cells are sitting in a photo session and the cameraman is sitting here. It is almost like that. So, this bigger one, which is called the kinocilia, and all these are the stereocilia, they are connected with a connector linking the tip link. You could read that tip link out there, and the tip links, and here you have another set of links, okay? And these are all eventually connected to this cell. So, if this kinocilia moves on the left, then the rest of all the connectors will move to the left. If the kinocilia move to the right, then everything will move to the right, depending on, now if you look at this image, if depending on in which direction they are moving, it decides that what kind of current it is going to conduct, whether it will be inhibitory or excitatory. Okay. So, from here, so this is the molecular architecture. So, this is all consist of different kind of myosins, if you could see, and please I request you go through this reference. So, these myosins are basically they are also called the myosin motors. These are the molecular motors or the protein motors what is involved, and they are actin. So, if you those of you have, please go through the basics of actin myosin integration in order to results in the movement of the sliding movement. So, you are having these actins and the myosins, you have the adaptor proteins and lichen and so forth. So, this is how the molecular architecture looks like. It is a very complex and beautifully knit structure if you look at it. So, you see this wonderful supercoiled proteins which are forming the base or the framework, they are connected with each other, and they have a whole range of subtypes, and this is the schematics. Again, for the schematics you have to refer to this journal, that Journal of Physiology, D. N. Furness's article. So, if you look at this, this is where the channels are sitting. So, if they pull in one direction, the gate opens and the calcium enters. So, if you look at this micrograph and look at this micrograph very carefully. Okay. So, this is how the calcium entry is taking place. So, there is a pull. So, it is almost like a hinge of the door. There is a pull on the left, it pulls on the left like this, and the channel opens. This is how this structure functions. This was, while I was drawing it, it was really tough for people to visualize exactly what is happening. That is why I decided that I will give you a special lecture where I will have all these figures, the schematics, which will help you to appreciate how this whole process is taking place. And followed by this, if you come to, so these are called MET currents, okay, mechanotransductioning current basically. Okay. So, if you come back to this one, so this is where the current is being generated. You see this green patch, so that is showing that there is a calcium influx or the calcium current, and this is how you are recording those currents in terms of the nanoamperes. And if you go back to the previous notes, you will see, and these are the different deflections which are taking place, delta x showing in terms of the micrometer, the deflections which are taking place, and these are the currents which are generated with respect to the different level of delta x. So, this is the function. So, x axis is showing you the delta x, or which is the deflection, and on the other hand the y axis is plotting the current. Okay. So, here also you can see the different currents at different time points. So, this is how all these nanoampere currents are being measured, and these are very small currents, but good enough to code a piece of information what is being processed by the hair cells. Okay. So, coming back, so these are the hair bundles and the mechanotransduction what is taking place. Okay. So, as I was telling you, it is pretty much the repetition of it. So, there is a stimulus, they move in one direction. So, you see in this situation, this is all moving towards the left. Once they move towards the left, the calcium channels pulled and calcium channel opened, and again there is an adaptation and they come back to their original position. So, if you follow this diagram very carefully, then the whole mechanotransduction will be very clear to you people, and this is how they are attached. This is believed how they are attached. So, there is a spring, there is a tip density if you look at it, and this is that spring which is pulling. This pink color spring attached with a partly green color, pink plus green color this thing. So, this is what is pulling. So, if this one moves on the left, so automatically this will make the whole structure move to the left. Okay. From after giving you this overall outline, I will move on to the cochlear implant. So, how the cochlear implant really looks like. As a matter of fact, it was the cochlear implant, as I have mentioned earlier, it was the first one which was the success story of mankind in terms of neural engineering or neural prosthesis. Okay. So, this is how the cochlear implant works. You have a microphone, there is a sound processor, there is a transmitter, and there is a receiver. And from here, this signal is fed into the cochlea. In the cochlea, these are connected. This whole thing is connected to the cochlear nerves, and this cochlear nerves carries the signal to the auditory cortex. So, essentially what you are doing, you are ruling out, say for example, if these cells of yours, these hair cells of yours are all damaged. So, what you do, you fed the direct information, the sound information directly to the cochlear nerves, and the cochlear nerves and take the whole information through the cochlear nucleus to the auditory cortex of the brain, and where it is being processed. So, you are bypassing the whole cochlea, and this is what I wanted to highlight in one of the previous lecture. So, this is how it looks like, the cochlear implant. The auditory system is composed of outer ear. This is the summary of it, and the middle ear, and the comprising the tympanic membrane and the auditory ossicles, and the inner ear composed of the snail-like cochlea containing sensory hair cells bathed in a fluid. So, this was what we discussed. Okay. These sensory hair cells activate the fiber of the cochlear nerves, which emanate from the spiral ganglion cells and project to the cochlear nucleus of the auditory brainstem. The neural pathway that leads to the higher auditory processing center of the brain, the inferior colliculus, medial geniculate nucleus, and the primary auditory cortex. So, if you go through this, then this will give you an idea about how these signals are being processed. And if you read through here, so what is happening is that a CI consists of the cochlear implant, CI stands for the cochlear implant, consists of an external microphone which collects the sound waves. So, look at it, which collects the sound waves, and so this is what is collecting the sound waves. Okay. And a speech processor which converts the sound waves into electrical impulse. So, here is the speech processor, sound processor. Okay. Then next come which converts the sound wave into electrical impulse, and then transmitted to a receiver implanted under the skin. Here is the receiver which is underneath the skin. Okay. So, after the receiver implant, the receiver sends the electrical impulse to the microelectrode array implanted within the cochlea. So, here within the cochlea, the microelectrode array which are, so all these MEA what we have studied from the beginning. So, this is where they are finding their applications, why the MEA has to be studied in order to understand how this whole cell electrode interfacing is so very important criteria before all these kind of implants will be successful. Okay. Now, coming back to the slide. So, this is where microelectrode array implanted within the cochlea, the electrodes directly stimulate the correct population of auditory nerve, so that electrical signals are propagated to the appropriate tonotopic regions of the cochlear nucleus of the brain stem, and then onto the higher auditory cortex. So, from here this is going to the specific part, and from here it is moving to the auditory cortex. So, this is the overall summary of the cochlear implant what I wanted to show you people. Okay. From here, this is another cartoon which is showing the same thing, the microphone, the amplifier, the sound processor, the sound signal are converted into electrical signal, and then through the cochlea. So, this is where the cochlear nerves to the medial geniculate fiber or the thalamic region from the brain stem. From here it is moving to the enlarged field potential amplitude in the auditory cortex and enhanced synaptic currents. So, this is the situation when your hair cells, or what I showed you, those wonderful mechano sensing cells are getting degenerated, and you have no other option but to resort to neural engineering techniques in order to restore your vision. These are the extreme situation, but this is one of the big success story of mankind of last 150 years of man's quest to interface machine with its body parts. Okay. So, from here the stimulating hearing auditory neural pathway from the cochlea of the inner ear to the auditory cortex of the brain. So, hearing can be restored in the congenitally deaf kittens. This is where it was done with an electrode that stimulated the auditory nerves in response to environmental sound relayed via microphone and sound processor. It is pretty much the same thing what we just went through. So, electrical discharges of the neuron in the auditory pathway results in a strengthening of intracortical synapses. That was the synapse strengthening, if you look at it, enhanced synaptic currents. This is what it meant. Okay. From the results in a strengthening of the intracortical synapses and an enhancement of the neuronal response to sound. The area of the auditory cortex over which electrically evoked field potential can be registered is enlarged in cat with early implant red and yellow compared to the deaf cat, which is in blue. Okay. So, this is how it looks like. So, here is the blue showing you the deaf cat, the red and yellow are showing in the implanted cat. Okay. So, this is what I wish you people go through bit carefully. From here I move on to the next one, which is the eye or the retinal or visual or retinal prosthesis. So, this is I have already discussed with you people the structure of the eye and give you a part of the idea about how you place the camera in front of the eye. So, here I decided I will show you some of the real time pictures which will help you to appreciate this subject much better as compared to just me drawing out here on the screen. Okay. So, just the same way as we have walked through with the hearing or the ear prosthesis, I will walk you through with some wonderful pictures out here that will help you to understand it better. So, this is the overall, this I have already discussed with people. So, the structure of the eye, the cornea, the lens, and here you have the retina, and here you have the vitreous humor. So, the light travels all the way through, and I have already told you that there could be either a corneal blindness, where basically we say that if somebody has donated eye, basically it is the cornea which is being taken out from an deceased individual and implanted into another individual, and the cochlea has very little blood supply. So, the compatibility of the tissue, biocompatibility or immune reaction is very least. Then comes the blindness of the lens, which is essentially the cataract. So, you replace this lens with a synthetic lens, and now comes the retina, where if there is a damage in the retina, then we need something a little bit more than cornea or lens implants. Okay. What is that? And that is what we are going to discuss. So, this is the overall structure of the layout, the pigment epithelial cell, the rods and the cones layer. Then you have the horizontal cells, the amacrine cells, the bipolar cells, and the ganglionic cells. So, this is the complete layer which is dealing with the whole eye prosthesis, the whole structure of the eye, and this is in a much more kind of blown up picture, where you have the pigment epithelial cell sitting at the base. On top of that, you have these violet and black shade rods in the cones, and then you have the horizontal cells, then you have the bipolar cells, here you have the amacrine cells out here, go through it, and here you have the retinal ganglion cells which are carrying the message to the brain, and this is how the light falls. Light falls from this side actually, as a matter of fact, always remember light is falling. So, essentially here the first layer, what you see, this red could be divided, the first layer is the retinal ganglion cell layer, but since neither of this layer or this layer L6, L5, L4, L3 has any kind of photoreceptors or any kind of light sensing proteins in them. So, the light is only sensed by the photoreceptor layer, okay, and beyond that there is an opaque layer of the pigment epithelial cell, which is essentially the the back of the eye. If these two lines, so this the front is retinal ganglion cell, and the bottom will be the pigment epithelial cell, okay, which is basically helps in having a cushioning effect. So, this is how physically a rod and a cone looks like. These are from the fish retina, and the reference is given here. Please go through this if any one of you is interested really to see how they look like and how they are being isolated from the fish retina. So, it is a paper in comparative biochemistry and physiology part A in molecular integrative physiology in the year 2008, in the month of August. This is volume 158 and issue number 4. So these are the rod cells. Physically, that's how they look like. This is where the outer segment of the rod cells. This is the inner segment of the rod cells. This is where all the light sensitive proteins, rhodopsins, are sitting. Okay, and this is from where the impulse is being sent. So all the dark current which are measured, it is measured here. Similarly, here is the cone. So this is the outer segment where the specific rhodopsin are sensing RGB, red, green, and blue color. They are sitting here, and then out here, from here the signal moves. And look at the dimension. They are in, even their thickness is less than 10 micron. Okay, so this is the kind of dimensions we are talking about, and in the length they will be hardly 30 microns. Okay, so the rod system has low spatial resolution, but is extremely sensitive to light. So it is specialized for sensitivity at the expense of resolution. Rods do not discriminate different wavelengths of light. Okay, conversely, the cone system has very high resolution, but is relatively insensitive to light. That thus it is specialized for visual acuity at the expense of sensitivity. Cones can discriminate different wavelengths. Always remember this. Okay, now this is even a much more kind of almost giving you a three-dimensional look about the rods and the cones, and this is the reference from Progress in Retinal Eye Research published in 2009 in the month of July, volume 28 and issue number 4. Please go through it. I mean, these pictures, the whole reason to show you these pictures, that will give you an overall idea or a bit of an appreciation about how wonderfully all these different kind of structures have evolved over a period of thousands and thousands and thousands years of long drawn evolutionary time scale. Okay. Now coming back, so this is how the rods and cones are distributed. So distribution of photoreceptor in the eye. So please, again, you should go through this Progress in Retinal Eye Research in the year 2009. So distribution of the photoreceptor in the eye. Overall, rods outnumber the cones by a ratio of 20:1 or greater in the retina. However, in the fovea, the cone density is the highest and is correlated with visual acuity. So this is the foveal region. Okay. So these are those are the different areas of the retina. So fovea is rich in your cones. Okay. So now moving on to the different kind of currents which are being measured. So rods are highly sensitive. So again, this is from the Comparative Biochemistry and Physiology Part A of the Molecular Integrative Physiology, the year 2008. Please go through this. So rods are highly sensitive, light sensitive, but their flash response time course is slow, so that they can detect a single photon in the dark, but are not good at detecting object moving quickly. Okay. Cones are less sensitive, less light sensitive, and their flash response time course is fast, so that cones mediate daylight vision and are more suitable to detect moving objects than rods. This is a very critical thing which I wish to highlight, and this is what is called the flash response time. So the flash response time of the rod cells. So something moves like this, the cone can detect it, but something like this, a cone can figure that out. Rods won't be able to, because they need bit of a time to adjust and understand a flash of light. Okay. So this is something very, very interesting for you people to really look at carefully. That so if you look at the slide again bit carefully, you see the time window. What is most important here? Look at the time window, the response time. How much time it takes for each one of them to respond. Okay. So after this, so this is about the cones, all the different cones, the blue cones, the green cones, and the red cones, and the different wavelengths they could perceive. This is how, and there are several species which have different other wavelengths which they can perceive. Okay. The fishes could perceive another color. Okay. So this is how, once they are isolated in the cell culture, this is how they look like. All these are the rods. You could see these rods, they are rod like, and they are a bit more purified. Here you have the rod cells, and here you have the cone cells. Okay. this is how they look like in a culture dish. So that's why I wish you people should have a very clear idea on how they kind of look in the culture dish and how they grow from here on. Okay. So vertebrate photoreceptor hyperpolarize on light stimulation was of great surprise to the neurobiologist, because it was an exceptional case that a neuron shows hyperpolarization, not a depolarization on stimulation. So this you won't be able to read, just note it down. Hyperpolarization, not a depolarization on stimulation. Okay. So this is where I was trying to show you the dark currents. You remember I was telling you about the dark current. So this is the outer membrane, and within the outer membrane, this is how all those different proteins which are involved in this, phosphodiesterase and cyclic GMP and all those things. So this is the disc membrane and this is the outer membrane, and this is how they are arranged out here. Okay. So and this is the picture of how the dark current are being measured. So you have the electrode in the presence of light. In the presence of light, what you see is the minus 70 millivolt out here, and where there is darkness, you see minus 40 millivolts. So essentially, it was that whole concept of dark current what I have introduced you, and what exactly is happening. So when the light is binding to this membrane, so what it does, so this is the, one second, let me, so this is what is happening when the light is binding. So this 11 cis retinal which is present there along with opsin, which is forming the complete rhodopsin moiety, split up into, so the first thing happened when the photon goes, this 11 cis retinal become 11 trans retinal, and then 11 trans retinal detaches from the opsin. So this is the detachment, which is also called bleaching. So then what would happen, this 11 trans retinal through an enzymatic process transformed into 11 cis retinal, but in the meantime the opsin does certain few other things, and that will be coming what opsin is doing in this slide. Opsin activates a protein called transducin, and the transducin activates something called phosphodiesterase, and this phosphodiesterase remove the cyclic GMP gating of the sodium channel, which ensures the entry of the. So if you look at the slide very carefully, you will see the cyclic GMP level declines and the gated sodium channels closes. So this is step 3 is very important for you to look at. Okay, this is that is step 3, which is very important. So this is where phosphodiesterase is binding and removing this gate. So if you look at this picture, it was a cyclic GMP which is holding this sodium channel up here where I am circling. Okay, so once the light falls, what happens is this cyclic GMP out here is being removed from that binding site by phosphodiesterase, and the phosphodiesterase removes it, that this channel closes, and once this channel closes, the current flow through the sodium channel stops, and this is how the signal transduction within the eye takes place. Now this is a much more complex picture. What is happening? Light is falling, rhodopsin is photoactivated, transducin activated, followed by phosphodiesterase activation, hydrolysis of cyclic GMP decrease in the cytosol, then the sodium channel close and the signal moves. So this is what is the light cascade. The first is, this is the basic overall structure of the rod cells, followed by a photoexcitation, what is taking place where 11 cis retinal is transformed into 11 trans retinal, and this leads to the bleaching process, and then comes the what is happening in the rhodopsin moiety, all the arrangement and the rearrangement within it. Okay, cyclic GMP as transmitter of vision. So this is another cartoon showing the photoreceptor, and so this is something very interesting which I haven't highlighted previously. The photoreceptor and bipolar cells have no action potential. They do not really shoot action potential. It is only the current which is generated which moves on to the next one. Hyperpolarizing membrane potential generates that current which moves on to depolarize the bipolar cells. Okay, and then followed by the ganglion cell where the action potentials are being generated. So this is how the signals are moving, if you follow this very carefully. So please remember, photoreceptor and bipolar cells have no action potentials. Hyperpolarizing the membrane potential, then leading to the depolarization of bipolar cells, and then at the ganglionic cell layer is all the processing taking place. So, essentially what we meant by that, if you look at this picture, go back. So, all the action potentials are generated in this layer, in this layer at the L6 layer, retinal ganglionic epithelial cell. Okay. And coming back to the, and so this is another picture where we are showing the phosphodiesterase is sitting, which is converting the cyclic GMP into GMP and thereby closing the sodium channels. And if you look through it, this is how the phosphodiesterase is sitting quiet. It is unable to act till there is an activation from the transducin protein, and this is how they are arranged along the membrane. So, these are all G protein coupled systems, and this is the summary of it, the bleaching and the regeneration of the visual pigment, where it is again getting regenerated, and again the 11 cis retinal, which is through an enzymatic process from trans to become cis, and the cis again gets incorporated into the opsin moiety and again gets back to the membrane. Okay. So, now coming to the implant position in the body. So, this I have already discussed, but now I wanted to show you the exact picture. So, this is where you can put the implant. Either you can put something out here in the front of the eye, and this is how it looks like that. So, this is the cornea, this is the lens, and this is where you can put. You can put an implant here. Either you put something in the front of the eye, a camera, or you could have an implant here where you see this C. Okay. So, here is the retina, here is the choroidea and the fixation of the cable at the exit point. Okay. Then you have the transscleral transcordial entry and the sclera. Okay. You can do it on the sclera. Okay. So, this is how it will work. So, this is the implant, actual implant, which is sitting out here. This is where the light is falling, the light sensitive micro photodiodes, then you have the amplifiers which are sitting there, you have the DS electrodes. Okay. And from here this signal is being sent all the way to the brain. So, please go through this reference by Zrenner E. You could see this in the Proceedings of the Royal Society, Part B, which was published in the year 2010. Okay. Please go through, please mark this reference and go through it. So, now this is how most of those implants look like. These are the complete. So, these are basically the light sensitive micro photodiodes which are sitting there, and this is where the image plate is involved and all the electrodes are connected underneath. So, you have the the photodiode electrodes and the contact holes. This is how they look like physically. Please go through this reference, that will give you an idea about how they really look like. And these are the different spots where you can really make all the connections. And there is a reference which is given, Expert Review in Ophthalmology, published in the year 2009. So, PS is the power supply, R is the receiver, S is the stimulator, and SP is the signal processor. So, this is how these different cameras are being put and different implants are being placed. So, there are different regions where they are being placed. Okay. So, and if you look at the summary of it, please go through this summary of the different concept of intraocular retinal prosthesis, and this is either it could be a epiretinal concept or it could be a subretinal concept. So, these are those detail which those who are interested really to understand the different systems. So, there is something called a Boston retinal implant system, please go through it. Then there you have intelligent medical implant systems, then you have the second sight Argus system. So, these are the different people who have over the years have kind of developed these different prosthesis regime, and they are the ones who have come up with different models in order to for people to understand that how these different prosthesis takes place. So, what I wanted you people a kind of a take home message from this is, though I mean this is a very tricky and a challenging area in terms of the fact that, as I am repeatedly telling in this course whenever I am talking about animal electricity, that how successfully we can implant an electrode without really disturbing the cellular assembly where we are doing the implant. There will be of course some degree of disturbances, but how we could minimize the immune reaction, how we could ensure over the period of longer period of time, the electrode fidelity will be maintained. These are some very challenging problems which mankind is going to face for in the years and centuries to come, because this is a long run thing. This is not something like, it is possible. It's how much we can fine tune, how far we could be innovative enough to take it to that level where these becomes a routine thing. So it's a journey, and please go through these references, because after this, once I have done these two processes, I will be moving on to about the man-machine interface, where people have tried really to implant train monkeys. I'll be coming to that after this. So I'm closing in on this one. So please go through these references and try to philosophize it in your system, because it is something which you have to think a lot. This is not something, because we know what could be done, but what are the innovative ways. Okay, thanks a lot.